All right. So.
Let's wait for it.
Oh, wait.
You have to start at 5 past.
5 past. Okay.
Second talk of the session. Second speaker is Fabrizio Costa. BlockClust, classification of uncoded RNAs.
Thank you. All right. So it is nowadays well understood that non-coding RNA do play an essential role in the regulation of cell. And however, out of, say, hundreds of thousands of predicted non-coding RNAs alone in the human genome, very few can receive an annotation, a functional annotation. And so in order to address this state of affairs and try and develop pipelines that can help us put order and identify automatically functions in non-coding RNAs, one has to proceed, for example, with non-supervised methods, for example, clustering. And but everybody knows that function of non-coding RNAs is both dependent on the sequence information on one hand, but also on the structure information. And so the clustering has to be a clustering that takes into account both this type of information, so the sequence and the structure. Now, there is a problem with that because while we can have experimental determination of the sequence, so far experimental data for the structure is not available in the quality and the quantity we would need. So structure is actually predicted. So we resort to computational methods to predict the structure, the folding structure of RNA. Now, this is, however, affected by noise. For one reason, once we have a sequence that we can then map to a reference genome, we cannot rely on this on information from the reference genome because they do happen upon transcriptional modification. So the sequence is not exactly the one that really lives in the cell. There are some non-coding RNAs that do not possess an intrinsic stable and conserved structure, for example, long non-coding RNAs. But even for short non-coding RNAs that do have a fairly stable structure, we do have only approximate energy models with which to build our structural hypothesis. For example, most of the times we don't take into account pseudoknots or higher order interactions. And so necessarily the structure that's predicted is not is affected by noise. Moreover, there's further sources of noise. For example, we at times are not sure of the exact boundaries of the transcript, and therefore we have to resort to a more local type of structure prediction, and local structure prediction is actually even a harder task than global prediction. So there are a lot of issues that affect the quality of the structure for non-coding RNAs. That's going to hinder our effort for clustering. So how about trying to address the problem of estimating similarities between non-coding RNAs using a different source of information. So instead of relying on the genomic sequence so the sequence information available from genomes, or the predicted secondary structure? How about using a notion of similarity based on profiles of read counts? So based essentially on the pattern of processing and the traces of this processing that is actually detectable from small RNA-seq experiments. So it is known, although not very well understood, that mature non-coding RNA is actually sliced and subject to being fragmented into smaller pieces by the machinery, enzymatic machinery of the cell. And it is believed that these fragments, they serve other purposes. They serve, they're then used for regulatory, to regulate the cell. So it is known, for example, for tRNA that there are enzymes that cleave it where the loop regions are, and so you obtain several fragments. The first, the 5', the 3' quarter, or the 3' half, or 5' half, and they do have different functions. So this is a widespread mechanism, and we can try and make use of that and see how the notion of how many of these fragments are detectable, if we can use that as a signal to infer what was the original non-coding RNA, to characterize non-coding RNAs. So we propose a framework that is based on two key notions. First notion is that we encode all the data that we have and the knowledge that we have about the problem into a rich format, into a graph. We don't have to, think hard to construct a sequence or encode in a sequence format our information. We are free to express all that we know in a full-fledged graph format. And the other key notion is that we then employ a fast graph kernel to then build predictive models or go about for our clustering. Now, this type of framework has been used in our group quite successfully for a wide range of applications. So we started in chemoinformatics QSAR applications for predicting toxicity. We showed how we can cluster hundreds of thousands of structures from RNA in matters of hours. We have shown how to model protein-peptide interactions. We've seen that this type of approaches are very reliable for predicting RNA binding protein target sites. We've even used it for predicting the direction of CRISPR arrays, and we'll show how to predict the microRNA mRNA interactions. So this type of framework has two key advantages. So the first one is in expressivity, and the second one is in efficiency. Because you're modeling entities as nodes and their relationships as edges, this gives a sort of universal encoding capacity. You can really model anything you're dealing with. Now, the key point, though, is that to this huge expressivity, it is not associated untractable runtimes or complexity. In fact, we show how we can convert these graphs, these discrete structures, into a sparse representation, and from that obtain classification methods and clustering methods which are as efficient as it gets. So they run in linear time, both classification and clustering. So now I'll go concretely into how we implement this method for our RNA-seq data. So the idea is that we start from profiles, read counts, and then we'll make a vector representation, and then we'll make a model. So we start from the reads, we have them aligned, we convert them into tags so that we actually count per position how many reads we have. From the tags, using the BlockBuster tool, we extract blocks. So the tool fits a Gaussian and then identifies a coherent block. And from these blocks, then we consider sequences of these blocks as our descriptors of our non-coding, candidate non-coding RNA sequences. So we have sequences of blocks that contains these read counts, these profiles, and we extract some measurements, some attributes from per block, which we then discretize. And we convert all this discretized information into graphs. And then from that, we extract the sparse vector, as I'll show. So here's a sketch of what's going on. We consider attributes or measurements from the single blocks. So we look at the redistribution within one block. We also consider attributes that exist between relations that exist between blocks, for example, if they're overlapping or not. And we also extract measurements for the full whole sequence of blocks. Okay. The type of measurements of attributes that we're interested in range from, so we tried a bunch of them, range from the average value or the count value or the entropy of all the values of, for example, the start and position of the reads within a block, the expression, the counts, and so forth. For every of those attributes, we discretize them into equal frequency discretization with an equal frequency discretization approach. And we experimentally determined that really 3 levels are actually optimal. So we have sort of a small, medium, large kind of information. And so what you see in the picture there is for different types of non-coding RNAs, you see the typical values for in every box. There's a different attribute. So we have a bunch of them, and then we cross-validate and find which of the attributes are best for discriminating. So once we have all these attributes, we represent all the information that we have into a graph format. Specifically, we build this sort of comb-shaped graph. So we have a backbone where we represent all the blocks that we have. And then we actually represent the attributes as hanging extra vertices. Okay, so in all the vertices, it says 1, 2, and 3. This is the first attribute, second attribute, third attribute. So then we have a graph. And what we do then is, with a sort of a combinatorial approach, we extract features. So we decompose these graphs into small fragments. And specifically, the fragments that we use are pairs of small subgraphs at a short distance from each other. Now I'll show you an example. So every single such pair of subgraphs gets an ID, a code, and that's our feature, and then we have a sparse vector. So we start, it's not very visible, with subgraphs which are given every vertex. It's all the vertices that are reachable within distance up to 1, 2, or 3. And so we start 1, 2, and we keep on going until maximum radius. Not only we consider these subgraphs, but we consider pairs of these subgraphs that are nearby at a certain distance, say distance 5, and then we consider all possible pairs. So all possible distances from 0 to a maximum distance, and we consider all possible fragments. So all this can be officially computed. They can be given, it's a bit tricky, but it can be given a unique identifier. And then from that, we count the number of occurrences in one instance, and we get a feature representation, a vector representation, a sparse vector representation of our graph. With that, once we have these sparse vectors, then we can go along and do classification. We can create similarity notion and measure distances. As for example, we can cluster. If we have annotated RNA-seq data, where we know where the different types of microRNAs are, of non-coding RNAs are, then we can try and see whether the same type tends to cluster next to each other according to our similarity notion. So for example, we compare with other methods, for example, deep block line. That's a method that's still based on the notion of blocks. In fact, uses the same blockbuster tool to construct blocks, but then it uses full alignment of these sequences of blocks to identify a similarity notion. And we're quite competitive, so we have most of the results are above in the order of 70s or 80s rock value. That's when we, ideally you don't have it, it's unsupervised, you don't know what you're clustering. If you do know, and if you want to build models that are capable to detect specific families, for example, microRNAs or tRNAs, we can do that. And you obtain fairly good classifiers. Here we compete with Dario. It's based on a random forest approach that it's quite competitive. In fact, it does a bit better in the recall level. What's peculiar of the approach we're proposing is that by obtaining efficiently these discrete structures and then having this explicit vector representation is that we get very, very fast. So a whole RNA-seq data set can be processed in a few tens of seconds as compared to other methods that take several hours. So a very interesting result is that these models are very stable across all sorts of different conditions. So if you create a model on human RNA-seq data, and you try to apply this model on different cell lines for different organisms and different tissues, you still obtain a fairly good precision in identifying the different types of non-coding RNA. It was quite surprising. Also, this type of discrete structures are very compact. So if you start from 16 million reads, 1.7 gigabytes of data, you can reduce all this information once you discretize all the attributes and you only consider the blocks into nothing more than 6 kilobytes. And so you can manage quite a large volume. If you consider highly precise clusters, then you can look into a consensus profile, a read profile, to try and gain some insight in what's the typical look of such profiles. And you see that they are fairly distinct, and you can identify those profiles that are more responsible, that are indicating different classes of non-coding RNAs. Like here, microRNAs or tRNAs or snow RNAs. Okay, and so the code is available. The pre-compiled predictions are online. They're linked against the genome browser. The whole software is available as a Galaxy module, so you can download it and run the whole workflow. And with that, I hope I've interested you, and I thank you for your attention.
So, I'm going to give you a question. Okay, this is a random question, so don't worry. And it doesn't really get a genius to identify a microRNA.
Yes.
So I can just look at the length, and I know.
Yes.
So,
The problem is the novel non-coding RNAs. There's hundreds of thousands of non-coding RNAs. We have no idea what they're going to do.
I understand, but classes are small. So you can convince me by saying that, okay, you train it, and you can immediately say, tell the difference between 16S versus 5S RNA.
By the size, yes.
By the size you can do, but how about the patterns? Can you do some unsupervised clustering, and you start with some further subclass and emerge? Subclass and emerge.
So in this case, of course, so we don't get that all, for example, tRNAs or all microRNAs are clustered together. We obtain very, very many subclusters, and each one is specific for the processing. So as I showed before, there are these enzymes which are known to process tRNAs, and of course the way they cut it depends on the tRNA. So certain tRNAs receive, certain are decomposed in certain types of profiles. So it's interesting then to look into why this is so, but that's a more biological question. So now we were only interested if we can use this as a signal to identify similar non-coding RNAs. So we find all the tRNAs which are similar in a certain subgroup, and we hope we can find unknown clusters of unknown functionally active non-coding RNAs. But it's not a tool that wants to replace, I don't know, tRNA finders.
So if you apply this approach to discriminate normal versus cancer study, how does this go?
To discriminate what, sorry?
Cancer study. So if you have two samples of RNA samples.
Yes, we haven't looked into using it as, to identify markers for, say, cancer or not. But so the tool is out there, is available. It's extremely fast, so it should be relatively straightforward to use it.
You haven't tested yet?
We haven't tested it to discriminate or to find specific markers for illnesses.
So you don't even know whether certain patterns are tissue specific or not.
Yeah, because there are a lot of defragmented sequences and mutations, everything.
On the other hand, it's very robust across cell lines. So.
I have a really simple question. I think I might have missed something, but are you using a regular RNA-seq data, or are you using a small RNA-seq data?
Small RNA-seq.
Small RNA-seq
Yes,
data.
so we filter for size. I mean, the protocol we only look at, we don't look at mRNAs.
So it's based on a specific size selection protocol. So,
Yes.
for example, if you want to look at microRNA, it's usually between 18 nucleotides to 25
So now
nucleotides.
we're more tolerant. So we go for hundreds of nucleotides.
Up to like hundreds of nucleotides.
Yeah,
Okay.
and so we try to capture all non-coding RNAs, and then we try to cluster what we capture.